Hello everyone. Today we are going to study Wilson's disease, a very important topic in GI pathology, especially liver pathology, and this can be asked in your university exams, your PG exams, and even in your MCQ exams. So let's understand the pathophysiology of Wilson's disease. Before understanding Wilson's disease, let's explain in basic what exactly does it mean. So, Wilson's disease is a metabolic disorder related to copper. So, if it is metabolic disorder, then of course there has to do something to do with its metabolism, right? Yes, and Wilson's disease is an autosomal recessive condition. Now, according to Robin's pathology, Wilson's disease is defined as an inherited metabolic disorder caused due to failure of incorporation of copper into ceruloplasmin. Now, if you may ask what is ceruloplasmin, so this ceruloplasmin is in a way you could say that it's like a storage form of copper, just like how excess iron or additional iron is stored as ferritin. Copper is stored as ceruloplasmin in our body. So let's move on to understand the copper metabolism. And before explaining that, let's understand why is copper important to human body. So first of all, copper is a part of complex 4 in electron transport chain, meaning that without copper you cannot actually synthesize ATP properly. And second of all, copper is a part, or should I say cofactor, of multiple enzymes in our body. Multiple enzymes. Now, copper has a lot of other functions as well, but for now, understanding Wilson's disease, I believe that this many functions will be enough. So now let's move on to copper metabolism. So what happens in our body is copper taken from our diet. From there it reaches the enterocytes. Now enterocytes mean cell of intestine, particularly the duodenum and the proximal part of small intestine. Now what actually happens is once the copper reaches the enterocytes, there is a protein in these enterocytes called copper membrane transporter protein 1. Copper membrane transporter. Now this will promote the uptake of copper into enterocytes. So once the copper enters the enterocytes, there is a role of another protein called ATP7A. Now what this protein does is actually it will help to bind copper with albumin and release into circulation. I mean the blood, particularly the portal vein. Now if you ask why, because portal vein brings all the nutrients absorbed from the small intestine to the liver first, and then only it is distributed systemically as required. And now, so we have copper bound albumin. Now this copper bound albumin enters the liver. Let's draw liver here. So copper is now inside the liver. Now there is a protein called apoceruloplasmin. Now this protein binds with copper with the help of another protein called ATP7B, and this will lead to formation of ceruloplasmin, and this is our storage form of copper. And not only that, ATP7B also helps in excretion of copper, excretion of excess copper into bile, and eventually out of the body through fishes. So this was all about copper metabolism. So let's start again. So copper taken from our diet reaches the enterocytes, and in the enterocytes we have copper membrane transporter proteins. Now this copper membrane transporter promotes the uptake of copper into enterocytes. Now with the help of ATP7A protein, our with the help of ATP7A protein, our copper will bind with albumin and released into circulation in the portal vein. Now the copper is bound with albumin, and now this copper bound albumin enters liver. Now copper combines with apoceruloplasmin in the liver in the presence of a protein called ATP7B. Now this will lead to the formation of ceruloplasmin. And what is this ceruloplasmin? Storage form of copper. And this is distributed into various tissues, and then eventually, if required, it can also be excreted. Now what happens actually if there is a problem in this ATP7B? That's right, you get Wilson's disease. So let's understand, if there is a problem in ATP7B, then that means this copper won't be bound to apoceruloplasmin level, right? So it won't be bound to it won't form ceruloplasmin, and no ceruloplasmin means no storage form, and no storage form means it can't be distributed into multiple tissues, and there will be problem in excretion. So technically there is high levels of copper, but at the same time there is no mechanism to utilize it. So that's our Wilson's disease. Now what are the methods of diagnosis for Wilson's disease? One of the most commonly used methods for diagnosis is decreased level of ceruloplasmin. Now if you ask why, we have higher level of copper levels but no mechanism to utilize it. So the body will start using the present ceruloplasmin to compensate for inavailability of copper to be utilized. So ceruloplasmin levels will start decreasing, and another reason, this ceruloplasmin is not being formed. Initially it was being formed and being utilized, and that's okay, but now it's only being utilized but not being formed. So decreased serum ceruloplasmin is one of the most common biochemical methods for diagnosis of Wilson's disease. And another one is increase in copper. Like I said, there is increase in copper but not the form that can be utilized by our body. So increase in copper, specifically hepatic copper, and this is the most sensitive one. Now Another one is increased urinary excretion of copper. So there is increased level of copper in your blood, and eventually it will reach this renal circulation and eventually kidneys, and then it will be excreted. So increased urinary excretion of copper is one way to diagnose Wilson's disease. So what is the treatment options? The best treatment option is penicillamine. Now don't confuse it with penicillin, it's a totally different drug, but for now let's understand what does this penicillamine do. So we all have heard of chelating agent, right? So what this drug does is it binds to copper, promotes its excretion, and in this way reduces free copper levels in our body. Another one is zinc. How does zinc act is it minimizes or reduces copper absorption right from the start. So these are the two drugs used in Wilson's disease. And let's understand another use of zinc, right? We heard, let's divert from the topic a little, and we know that this is used in diarrhea. You know why? Because it reduces both severity and frequency of diarrhea in children. We do give it to adults as well, and if you want to understand dosage, so for children less than 1 year suffering from diarrhea, we give them about a 10 milligrams per day maximum. For greater than 1 year, we can give up to 20 milligrams per day. So I hope you have learned something with this video, and if you would like to see me make a YouTube short video for summarizing all of this in 1 minute, then do comment down, and I hope you've loved my explanation. Do provide any feedback you need, and thanks for watching.